Biology chapter 19, theory video 1, genetic technology is our chapter. So gene technology is it has many different uses. For example, in forensics, finding out who has committed murders or whatever the crimes, or whatever the sample that is. So we can actually using some bodily fluid, we can figure out, or we can relate the criminal to a certain place or a instrument or whatever it is. So this is all done by the DNA, by studying DNA. So we have had the breakthrough phenomena developed during the 1950s and 60s in relation to DNA. So that DNA technology has been changed so much during the past few years that we can actually change DNA and also change all of the proteins. So the DNA has a coding for the production of proteins. So we can change the proteins which are synthesized by the proteins, by the DNA, and also as a result of the cell. And we can study the nucleotide sequences, we can change them, and also we can carry out genetic tests to identify if they are carriers of genetic diseases. And also we can do gene therapy or gene editing to those who have these conditions. So, and also in food technology, there are many different advancements that have been made by the changing of the genes. However, there are some social and ethical issues that we have to face when dealing with genes. Now, genetic engineering is the uh, is the is the introduction of a new gene to an organism from another. That is to remove a gene from one organism and transfer it to another, so that gene is expressed. So then we say that the DNA that was there in the original original organism is changed, uh, and uh, now it will contain lengths of nucleotides from two different organisms, and these are known as rDNA or recombinant DNA. So the gene that is expressed, the organism in which the gene is expressed would be known as transgenic organism or genetically modified organism, that is GMO. So the DNA would be from another species, entirely a new different species, or or a different organism from the same species. So we can actually now here what we are doing is we are overcoming the barriers that are there for genetic transfer between species, or else if there is an organism with a uh, organism with a quality that is advantageous, and if there is no other way than introducing the gene, then we can do that. For example, we can insert a human gene into a bacterium. So that is how we produce insulin, uh, by giving our genes to the bacteria. So here, when we actually speak about genetic engineering, it is a result of the transfer of a single gene. This is different to selective breeding. In selective breeding, we speak about the entire set of genes that are being involved in developing that uh, uh, individual organism. So to have an overview about the genetic transfer or gene transfer, we have to talk about genetically modified organism. There are different ways in which a GMO can be produced, but these steps are going to be essential part of it. The first one is the gene that is required should be identified. it should be cut out from a chromosome, or it could be uh, made from mRNA by reverse transcription, or it could be synthesized from nucleotides. So one of these three ways we have to identify and obtain the required gene that we need to be expressed. Then multiple copies of the gene should be made using technique called the polymerase chain reaction, and we discussed about that earlier as well. So using the polymerase chain reaction, we produce different copies of the gene that is needed. Then the gene is inserted into a vector. So the vector is not involved in the in expressing the thing by expressing the quality, but is involved in delivering the gene to the cells of the organism. So these vectors could be plasmids, viruses, or liposomes. Then the vector will take the gene into the cell. Now the cell that have the new gene are identified. We can use marker genes and then we to identify it, and then we clone these new cells. So the to perform these activities, that is to perform the genetic engineering thing, there would be a toolkit that would be needed by the genetic engineer. First one that he would need is the enzymes, the restriction endonucleases, DNA ligases, and reverse transcriptase. Then the vectors, including plasmids and viruses, would be needed, and the gene coding for easily identifiable substances that can be used as markers. So they would need, the genetic engineer will need a toolkit consisting of enzymes. The enzymes would be restriction endonucleases, DNA ligases, and reverse transcriptase. Then the vectors would be the plasmids and the viruses. Then gene coding for easily identifiable substances that can be used as markers. So these are the tool. This is the toolkit that is required by a genetic engineer. Write down a note on this. Genetic engineering. When gene technology is taken into our advantage, there are multiple uses, including development of genetically modified organisms, who would demonstrate qualities that we require. This is done by expressing the genes which would produce proteins that are useful for us. Are you line and right? Genetic information in one organism is changed by altering the base sequence of a gene, or by introducing a gene from another organism, is referred to as genetic engineering. And the organism thus produced is said to be a genetically modified organism, within brackets capital GMO, all capital. And the resulting DNA would be known as recombinant DNA, simple R, capital DNA. The organism which expresses the gene is known as transgenic organism or genetically modified organism. In genetic engineering, following steps are essential. Number one, the gene that is required is identified, may be taken out of a chromosome, made from mRNA by reverse transcription, or synthesized by nucleotides. Number two, multiple copies of the gene are made using the technique polymerase chain reaction, capital PCR. Number three, the gene is inserted into a vector which would carry it to the organism, which should carry it to the cells of the organism. Examples of vectors are plasmids, viruses, and liposomes. Number four, vector takes the gene into the cell. Number five, the cells that have the new gene are identified by using marker genes, and the cell would be cloned. The toolkit that would leave a line and write the toolkit that would be needed by the genetic engineer would consist of: number one, enzymes such as restriction nucleases, DNA ligases, and DNA transcriptase. Number two, vectors including plasmids and viruses. Number three, genes coding for easily identifiable substances that can be used as gene markers. I hope you understood. If you have any questions, please ask.